Eugenio, what are the tools for meaningful drug discovery? And how can we efficiently find a new chemical structure that will successfully cure a disease, given the enormous number of compounds that need to be tested?
I think that in the past there was this brute force approach where there were libraries of 3-4 million compounds that have been screened. I think this is largely now overcome by other techniques, of course. I mean, there are different aspects to have. There is one side chemistry. In my opinion, chemistry is somehow limited because the synthesis capacity of complex molecules is not definitely easy. Even if there are interesting development, for example, use of artificial intelligence and automated synthesis that now start to generate really interesting compounds that normally would not have been designed by normal chemistry, this can overcome. I am a strong believer. I like very much to work with natural compounds because I think nature is power in chemistry that we don't have it, but definitely, of course, then some of the structure are very complex to replicate in the synthesis laboratory. But I like, for example, to work what we call semi-synthetic compounds, so backbone coming from natural compounds that can be synthesized and then used for screening. But the chemistry has a large component in the discovery because at the moment it is the key element to get the good candidate afterwards, and this chemical space increase is definitely a key. Other key elements are how relevant are the system or the assay that you use to your human pathology. More and more we drifted to use of human primary cells or stem cells derived cells. For example, we like to work a lot with the phenotypic assay because we think that even if it's difficult on the target because it's a target agnostic approach, but it still resembles a system level of complexity in a cells. And I think that's important. A famous German scientist, Virchow, in Berlin, some last 2 centuries ago, was telling that every pathology is a cell pathology, and I do believe that it's still valid. At the end of the day, we have cell pathology that it just then go to the human pathology. So having the cell as a system, it's much more indicative. The challenge in this case then is to make the deconvolution for the target and the mechanism of action. But I think with the actual techniques, it's possible to do this kind of approaches, especially with the artificial intelligence and the data that are available now is really getting more and more feasible to have this approach. All this together might help to increase the output of the drug discovery, and not only the output, but also the translation to the clinical phases that is getting easier, hopefully. So you can discover side effect or efficacy level at very early stage rather than getting at the clinical stage and then discover that your molecule is not efficient or is not does a side effect. So there are many things that can help in this bottleneck, twist the bottleneck there.